Hi students, today we are going to be talking about microorganisms, basically bacterium and viruses. Okay, so bacteria I'm sure is a very familiar concept to you all when you have, like, for example, food poisoning. And virus, the most common thing will be the recent coronavirus or even just your normal day-to-day flu virus itself. So bacteria itself come in many different forms, and they are typically named with the form that they take over or they take after in mind. So for example, if it is in a sphere-like shape under the microscope, they are often termed to be a cocci. Okay, so you can see that over here, these names will end off with a cocci, but of course, you're not expected to memorize the names of it. This is just an illustration on the different shapes of the bacteria. Just keep in mind that they are colored for visual purposes and they don't come colored most of the time. Okay, so general characteristics of bacteria is that they are prokaryotes. So unlike animals or mammals that you see around you, we are what we call to be eukaryotes that you have learned in the earlier part of the year. Okay, so eukaryotes versus prokaryote itself. Prokaryotes, they do not have a nucleus, meaning to say that the DNA is not in the nucleus, whereas in human cells, for example, in all of the human cells, with the exception of red blood cells, we have got DNA in the nucleus itself, protected in the nucleus by the nuclear membrane. But in prokaryotes, because of their simplistic structure, the DNA is not bounded by the membrane. So we can see in this picture here, the DNA is actually free floating in the cytoplasm itself. It also has the presence of a cell wall here, okay, which is often attacked by antibiotics when we take it whenever there's a bacterial infection. Okay, now there are two types of your bacteria. They can be heterotrophic or they can also be autotrophic. So auto comes from the word automatic, so meaning to say that similar to plants, which are also autotrophic, these bacteria, they are able to make their own food. Heterotrophic, on the other hand, means that they require food and is the opposite of autotrophic. So likewise, humans, we are heterotrophic because we require the intake of food. Okay, now typical bacteria here, it has got a flagellum here, so it enables the movement of the bacteria itself. So bacteria movement by the flagellum, we call them motile bacteria. Okay, so bacteria, they reproduce through binary fission in an extremely rapid manner. So we will look at what is binary fission in the next page itself. The DNA is floating. That is one difference in bacteria. Another thing that is unique to them is what is called plasmids. Plasmids, they are used for genetic engineering. So things like, for example, the production of insulin in modern day society for people who are diabetic. Okay, so when bacteria, they are heterotrophic, how do they actually take in the food? Okay, so they are either under the category of saprophytes, whereby they feed off dead and decaying matter, or eating parasites itself. Okay, so how they feed off the parasites is these parasites, they live in another organism. So for example, living in a human or in an animal. So you can see that we have got this cell wall at the exterior, which acts as a protective layer. Take note that this cell wall is not of a cellulose nature. Cellulose cell walls in plants is very different from the cell wall that is present in bacterial cells. So this cell wall is often targeted whenever someone is sick and is given antibiotics. So in order for the bacteria to survive properly, it requires that cell wall for survival. So this antibiotics, what it does is that it prevents the formation of the bacterial cell wall. So by preventing it, the cell content is unable to form properly and bacteria is unable to multiply itself through binary fission. Okay, so the next page here, we will look at the binary fission. So binary fission, it dependent on a range of conditions. So things like pH, temperature, whether it has got the right and the correct type of nutrients, also the good amount of nutrients. Presence of oxygen is more for genetic engineering. Okay, so genetic engineering is things like you have got insulin production in modern day society instead of removing the insulin from the pancreas of an animal. Okay, so over here we have got the one bacteria here. So you can see that the DNA is free floating. It is not enclosed by any nucleus itself. So the DNA doubles itself in the bacterial cell, and after that the cytoplasm increases in size, and you can see that the free floating DNA separates to two ends, and the division starts from the middle. So you can see that it is getting into a burrow in this direction, going downwards from the top, going upwards from the bottom, and these two areas will keep coming closer and closer to each other until it forms two complete different cell walls, and from there they divide completely to give you a new set of cells. So this daughter cell, we call it daughter cell, the new one that is being made, is 100% identical to the original cell. There is no change in the DNA structure. It is 100% identical. Okay, now some bacteria, they are very hard to kill because they form these things called endospores. So endospores, they are dormant, meaning to say that they can just lie inert, having no reaction to anything in the environment. So these are extremely hard to kill because you don't get destroyed by things like heat, UV, radiation, and disinfectants. So one example of this is by this Bacillus anthracis. Okay, this is the one that causes anthrax, which cause a lot of respiratory issues, etc. Okay, but of course not all bacteria are bad. There are also some good bacteria, things like, for example, in your Yakult that you drink to bring back the good health of the small intestines and also the large intestines. So the uses of these bacteria, in modern day, we have got things like the nitrifying bacteria. So this nitrifying bacteria is in the soils itself. So we have got bacteria in the soil which takes in nitrogen from the atmosphere, surrounds us, because after all, nitrogen takes up about 79% of the air. So these bacteria take in the nitrogen, convert it to usable nitrogen for the plants in the soil, because nitrates or nitrogen is used by the plants itself for growth and renewal of cells. Bacteria has also been engineered to use for sewage treatment. So what this bacteria does is that it is purposely made and engineered in the laboratories to break down the materials that is in the sewage, such as the carbohydrates and the nutrients in the waste. Okay, our new water probably uses this to make sure as one of the steps in recycling the water. Food production, things like your Yakult itself, fermentation, your cheese, yogurt, all this requires bacterial formation. Okay, antibiotic production, as we mentioned just now. Okay, so this antibiotic itself, it also uses things like fungus and bacteria to actually destroy other harmful bacteria. Okay. Genetic engineering is whereby we use one specific component in the bacteria called plasmids. So just now in the earlier page when we looked at one of the components here, it contains plasmid, okay, in bacteria. So this plasmid is then removed from the bacteria itself and used to produce insulin. More in-depth steps will be learned in the upper secondary level. So this one is just an overview of it. Okay. So next we look at viruses. So viruses is unique because most viruses, they require what we call a host. So which means it requires something that is living in order for it to survive. So replication only takes place in a host because viruses essentially, they are non-living things. Okay, so they are non-living things. They don't have a cell-like structure on its own. They are very unique, and which is also why they don't group under any of the main 5 kingdoms, such as your Animalia, your Plantae, etc. Okay, they are extremely small and tiny. They have got genetic material as well as proteins here. So you have got all these protein structures at the outside, which is able to bind onto different parts of the cells and therefore causing symptoms such as your flu viruses. So different types of viral shapes. Again, these are not colored in real life. These are just for your visual purposes. So all bacteria, they are parasites in nature because they cause harm to whoever have the virus itself. So the virus itself is this, unlike the bacteria which has got a cell wall, the virus is surrounded by a lipid bilayer. So lipid means fats, bi means two. Okay, so if you break up the word, it means fats in 2 layers, and this forms the virus envelope to actually protect the virus DNA itself, and also when it binds onto human cells or host cells, it increases the ability of these bacteria to infect the host. Okay, we have got some viruses that are very commonly known. Influenza, this is your common flu virus. SARS virus back in about 2003 or so, and HIV virus, which is very well known. So typically most viruses, not all, but most viruses can be so-called treated with vaccines. So we have got a virus that is injected into a human, for example, and once the virus in small quantities is injected, the human body will now be stimulated to produce the antibodies or to fight against this little bit of virus that is being injected. So your body now has a memory of this virus component, and that memory is through the production of the antibodies during the fighting. Okay, so the next time round, let's say we have got an actual flu virus that attacks the body. Now the body already have a memory of that particular virus, and therefore it doesn't have to wait to produce white blood cells in order to kill or destroy this flu virus. It can produce it almost immediately, and the symptoms will be less severe, if not none. So if you don't have a symptoms to flu, it means that you might have the flu virus, but it is just that the body is taking fast enough time to fight it all off, and therefore the symptoms are not portrayed. So some viruses have been used in two areas. One is for gene therapy. So using the virus itself, the gene is being transferred into the human body. For example, if let's say there is a gene deficiency or there is a gene mutation causing a severe effect on living conditions. Another thing is also the use of bacteriophages. So bacteriophages, if you break up the term, this bacterial comes from the term bacteria itself, and phages is a different form of a virus. So we have got a virus that attached to the cell surface region here, and once the virus penetrates into the cell, the DNA of the virus is injected into it. Okay, so right now the DNA is able to be injected into the cell, and the DNA content will now be multiplied. So once it is being multiplied, these viruses components can be assembled together into one complete viron, which is now called a bacteriophage, and it will then be released out to the environment. So we can use bacteriophages for multiple purposes, things like restoring the intestinal health, to destroy harmful bacteria. It is still a rather new area, but the results have been promising in this. Okay, so you see some differences between viruses and bacteria. Next, viruses is one of the smallest components in the world itself. Single cell organism. Okay, so for your viruses, these are single cell organisms. Bacteria, they are microscopic. You can't really see it with the naked eye, similar to the viruses. So bacteria contains your single chromosome. Single chromosome means one strand of DNA itself. Humans, we have got two strands. Okay, so bacteria has one strand. You have got a cell wall. This cell wall is not made up of cellulose. Okay, unlike your plant cell, it's a different type of cell wall. Consists of the typical cytoplasm, cell membrane, ribosomes, and enzymes. So you can see for viruses, it does not contain any of these things, unlike the bacteria, which is why it requires a living host or a living human or animal, so that it can hijack, it can take over your own body's machinery to replicate. Okay, so the virus itself will inject itself into your own cell and use your cell's nucleus, your cell's cytoplasm, your own cells, cell membrane, etc. to replicate itself. Okay, so beyond outside of its own territory, of its own host cells, these viruses, they are not able to reproduce. Okay, and because of that, they are considered to be non-living organisms. Okay. But for bacteria, they can undergo binary fission on their own. Okay, so independent production, they do need to eat. They can remove waste products, grow and reproduce, and therefore they are considered to be living organisms. Okay, that's the end of your bacteria and viruses. Hope it was good overview of it. You will learn more about it in your upper sec. Thank you.